What is going on, everybody? Josh here with Rise Above, back with a video today on the basics of the gut microbiome. What is it? Why does it matter? How does it change? How do we test it? I've been researching this and I'm going to cover all of that today. First, be sure to like, subscribe, and if you enjoy the video, leave a comment for the algorithm. Daddy needs a new pair of shoes. I don't, but if I wanted one, that would help. Now for the science talk. The gut microbiome is an intricate, multivariate balance of variety of bacteria, viruses, and organisms that interact in symbiotic, meaning mutual and or helpful, commensalistic, neutral, and pathogenic or harmful manner. There are a variety of species of bacteria in the gut microbiome, characterized by 11 different known phyla. The microbiome of every person on Earth is unique to some degree, and the balances of each microbiome vary. What is considered to be the gut, or the gastrointestinal tract, or GUT, gut stretches from the mouth all the way down through the esophagus, stomach, large and small intestines, and the colon. And each of these sections contain different combinations of bacteria. We are first colonized with these bacteria when we are young, as an infant, and particularly our first major incidence of colonization is when we are leaving the womb through the vaginal canal. In this case, C-section actually bypasses the natural colonization process induced by natural birth, and scientists believe this may lower the initial colonization of bacteria in infants. Breastfeeding is also considered an important early step in transmission of various bacterial species from mother to child. It makes sense then, children who are not breastfed are considered to be more potentially prone to certain illnesses. Other factors affecting transmission of microbes are your environment, the people and number of people you are surrounded by, as well as proximity to animals. The greater your exposure to a variety of microbes, the more likely diverse your microbiome will be. Of course, that is not to say go and lick a subway handrail. If you have a large family or live in close proximity with family members, or dogs, cats, etc., you're more likely to have a more diverse microbiome. Of course, what is most important is being able to determine the composition of a healthy gut microbiome versus an unhealthy one so that we can direct diet, lifestyle choices, and environmental choices in order to prevent disease and to promote health. You can see the multi-factor effects here on the microbiome. Studies are still being conducted to determine what a truly healthy gut microbiome looks like. Notably, the National Institute of Health is conducting something called the Human Microbiome Project in order to develop a standardized set of data along with models of what can be considered healthy or disease-causing microbiome compositions. Health is a multifactorial thing. In order for a human to be healthy, several factors need to be in place. Diet needs to be sufficient, nutrient-dense, and devoid of excess sugar and processed ingredients. We need to get adequate sleep and maintain a healthful metabolism and weight. Our microbes also need to develop over time and provide a sufficient balance of beneficial and commensal bacteria with a proper balance against the pathogenic bacteria. Healthful microbiome helps to instill what is called colonization resistance, which is the resistance to the overgrowth and colonization of pathogenic bacteria. In healthy people, pathogenic bacteria is unable or greatly reduced in its capacity to bloom or grow and cause diseases like colitis, among other things. Additionally, the gut is also the place where several very important neuromodulators and transmitters are produced in large quantities. So it is a fairly safe assumption to say that a healthy gut is very important to our neurological health. Things like serotonin and melatonin are both produced in large quantities there. With improper balances of serotonin and melatonin, you will have a very hard time sleeping and will likely become depressed. Let's do the general background on the microbiome. From a 2021 review entitled Nutritional Components in Western Diet versus Mediterranean Diet in the Gut. Quote, as previously reported, all domains of microorganisms compose the gut microbiota, including bacteria, archaea, viruses, fungi, and protozoa. According to the current evidence, the ratio between microorganisms and human cells is approximately 1 to 1. Bacteria are the major components of the microbial core. In fact, up to 2,776 species of bacteria have been recognized in the human gut, including 11 different phyla, like I mentioned. And forgive me, guys, I'm probably going to butcher the names of some of these phyla of bacteria. Firmicutes are the most extended bacterial family at 65%, followed by Bacteroidetes at 23%, and Actinobacteria at 5%. Next, a 2017 paper entitled Gut Microbiota, Role in Pathogen Colonization, Immune Responses, and Inflammatory Disease. Quote, in addition to the metabolic benefits, the microbiota provides the host with several functions that promote the intestinal epithelial barrier, meaning the gut lining, immune homeostasis, optimal immune responses, and protection against pathogen colonization. Some of the indigenous bacteria can promote disease under certain circumstances and are commonly referred to as pathobionts. The normal gut symbionts form a stable community that resists the invasion of non-native bacteria and the expansion of pathogenic biota. This phenomenon is known as colonization resistance and has been recognized since at least the 1950s. An immature bacterial community, such as in infants, or one that is disrupted by antibiotics, may lose this protective ability. Colonization resistance actually includes several related aspects: resistance to initial infection, improved tolerance of an established infection, and clearance of the infection. These all arise from the constant competition between normal gut residents. The mechanisms bacteria use to compete in the gut can be divided into two broad categories: direct and indirect. Growing evidence has shown that the perturbation of the gut microbial community may fuel blooms of otherwise low abundance and harmful bacteria, which can further exacerbate the intestinal inflammation. Certain types of E. coli have been observed in patients with Crohn's disease and ulcerative colitis, the two major forms of irritable bowel disease. You can see here in this image a healthy microbiome. There is a wide variety of different microbes. Beneficial and commensal bacteria work in harmony with the immune system to keep pathobionts, or unhealthy pathogenic bacteria, in check. The lining of the gut also remains intact, and there is little escape of pathogenic bacteria through the epithelial barrier, which could eventually make its way into the bloodstream. In cases of unhealthy gut, or dysbiosis, you can see a decrease in the diversity, and a loss of beneficial bacteria leads to an increase in pathogens, impaired antimicrobial response, and translocation, or escape, of bacteria outside the gut lining. By the way, guys, if you're enjoying this video and finding it informative, please hit the subscribe button and like the video. It will help others to find the channel. Let me break down the papers I found most useful in learning about the transmission of microbes to the microbiome. First, we'll start off with transmission, vertical and horizontal. For vertical transmission, a 2022 paper entitled Reconstitution and Transmission of Gut Microbiomes and Their Genes Between Generations. Quote, the newborn human gut is initially colonized via inoculation with maternal, vaginal, and fecal microbes when the baby passes through the birth canal, otherwise known as a vertical transmission. Direct vertical transmission during birth has been demonstrated in several animals, including bears, great apes, bats, mice, beetles, and fish, in addition to humans. Moreover, Lee et al. Demonstrated antibiotic administration to mothers during birth considerably reduced and changed the vertically transmitted species. Cesarean section delivery, or C-section, is one of the strongest disrupting factors of the normal colonization process and has been reported as a risk factor for disorders later in life. Human infants born by C-section are colonized initially and primarily by skin microbiota. Subsequent colonization occurs in humans by close physical contact of the offspring with parents, family, and community members by things such as kissing, hugging, and touching. Analysis of the DNA in several bacterial strains isolated from mother's milk demonstrated that they were identical to those found in their offspring. Fungi and viruses have also been detected in the breast milk of healthy human mothers, as well as the milk of other animals. Next, for horizontal transmission, the same 2022 paper, Court et al. Demonstrated that in humans, an average 10-second open-mouth kiss transfers approximately 80 million bacteria. 10 seconds. However, social transmission between individuals within herds and between populations, all being horizontal, also occurs in many animals, including non-human primates, herbivores, and birds. Reads, quote, offspring often acquire horizontally transmitted microbes, not from a parent, but from a family member or any other organism in which they are in close contact with. For example, it has been reported that humans acquire microbes from their pets. Although in some cases, horizontal transmission is apparently the main mode of transmission, mostly it takes place together with vertical transmission. Tung et al. Showed that social networks and social interactions in wild baboons could predict microbiome structure even after controlling for shared environment, diet, and relatedness. Microbiota that are absent from infants but present in childhood and adults are soon to be acquired by horizontal transmission, especially from family members. Horizontal transmission has a definite advantage, supplying functions that were not present in the initial vertically transmitted stock of microorganisms. However, horizontal transmission also occurs with viruses and pathogens, such as COVID, for example, or the bubonic plague, so it is not always positive. And finally, mixed mode transition, which is horizontal and vertical together, same article. Ebert claimed that mixed mode transmission is the dominant mode of transmission in the animal and plant worlds. So, with this information in mind, what does the research say about what constitutes a healthy gut microbiome? From a 2021 review entitled The Healthy Gut Microbiome, what is the definition? Quote, as studies have progressively included larger numbers of human volunteers, it has become clear that the microbiome variance among apparently healthy individuals is vast. No single microbiome configuration is required for health. Rather, various different configurations of the microbiome can be consistent with health. However, if health is considered a state in wait of the emergence of disease, then the concept of a healthy microbiome should be broadened to include microbial elements that protect and those which increase the risk of disease. Most of the variance within the human microbiome, as determined by population-based studies, is still unaccounted for. Meanwhile, it is clear that the microbiome, healthy or otherwise, does not exist in isolation. It is a reflection in part of host genetics, but mainly environmental and lifestyle factors, including ethnicity, geography, and diet. Because most people older than 50 in the developed world are consuming one or more daily medications, many of which are modifiers of the microbiome composition and function, medication history must be accommodated in any reference norm for the human microbiome. From a 2015 paper entitled The Role of Normal Gut Microbiota, quote, a recent large-scale study has estimated that the collective human gut microflora is composed of over 35,000 bacterial species. A Danish study of the gut microbiome and their function involves 123 non-obese and 169 obese individuals, resulted in the concept of a high gene count and low gene count, both of which have implications in health and disease. The high gene count microbiome includes a variety of bacteria. High gene count individuals have a functionally much more robust gut microbiome, lower prevalence of metabolic disorders and obesity. On the other hand, the low gene count individuals harbor a higher proportion of pro-inflammatory bacteria. The large intestine constitutes over 70% of all the microbes found in the body, and gut flora that is generally discussed in the context of disease states by and large implies the colonic flora, especially those derived from stool and metagenomic data. From a 2016 review entitled The Role of Diet on Gut and Microbiota Composition, quote, currently, 4 major microbial phyla are known to represent over 90% of the bacterial component of the gut microbiome: Firmicutes, Bacteroides, Proteobacteria, and Actinobacteria. The majority of quote-unquote good bacteria harboring the human gut microbiota are represented by Firmicutes and Cytophaga-Flavobacterium bacteroides, or CFB. As you can see from this research, having a highly diverse microbiome is considered to be one of the most important indicators we can find for health. A healthy microbiome tends to be more diverse and is mostly constituted by phyla Firmicutes and Bacteroides, with prominent representation of HGC or high gene count microbes. It's a bit sciencey, but just focus on diversity and higher representation of phyla, Firmicutes, and Bacteroides, right? That's a good starting point. Speaking of diet, since this is one of the key modifiers of gut health, let's look at the latest literature on what might be a good diet for maintaining a good balance and variety of gut bacteria. From a 2015 study, Role of the Normal Gut Microbiota, quote, changes in the composition of gut microbiota have been demonstrated among subjects according to different dietary intake. A particular diet may promote the growth of specific bacterial strains. Moreover, a high-fat diet can promote the development of pro-inflammatory gut microbiota, with a consequent increase of intestinal permeability. An important side note, it is very important to discuss what types of fats we're talking about and where they come from. A lot of these diet-related studies do not do a very good job of carving that out. Resuming, diet is known to have a strong influence on the composition of intestinal microbiota. To confirm this hypothesis, some researchers sequenced oral microbiota from skeleton teeth of people who lived over the various eras. They showed that the most significant changes in human gut microbiota has occurred during the two socio-dietary breakthroughs over the human history. The passage from the hunter-gatherer Paleolithic era to the farming Neolithic era about 10,000 years ago, with a diet rich in carbohydrates, and the beginning of the industrialized period characterized by processed flour and sugar diet about 2 centuries ago. Indeed, a high-fiber diet seems to positively correlate with bacterial richness. Therefore, permanent changes in gut microbiota composition might be achieved through dietary modifications. While Western diet, typically constituted by high consumption of red meat, animal fat, high sugar, and low fiber, was therefore associated with an increased number of Bacteroides phyla and also Ruminococcus. Specific changes in the gut microbiota have been associated with long-term and short-term dietary modifications. Wu and colleagues found that the microbiota composition is strongly associated with long-term dietary habits. Prevalence of Bacteroides and actinobacteria is positively associated with a high-fat diet, but is negatively associated with fiber intake, whereas Firmicutes and Proteobacteria show the opposite association. Also, short-term dietary alterations can modify. Consequently, the Western diet could therefore create the favorable conditions for chronic endotoxemia. Endotoxemia is associated with systemic inflammation with metabolic syndrome. Diets rich in fats and low in fibers, typical of Western countries, is rapidly spreading around the world. Similarly, inflammatory diseases are increasing in prevalence, even in areas where they were less common a few years ago. Between these observations, maybe there is a link which could be represented by diet. It is still too early to comprise whether this is only a suggestion or if there is a precise pathophysiological mechanism. Next, antibiotics. It is well known that antibiotics, while very useful for bacterial infection, also can have a major impact on the microfloral balance of the digestive system. A 2016 review entitled Interactions Between the Microbiota and Pathogenic Bacteria in the Gut reads, quote, antibiotics revolutionized medicine and were justifiably dubbed magic bullets against bacterial infections. However, conventional antibiotics are generally bacteriostatic or bactericidal, which means that they indiscriminately kill or prevent the growth of both pathogenic and beneficial microbes. Antibiotics can also alter the taxonomic, genomic, and functional features of the microbiota, and their effects can be rapid and sometimes everlasting. They can decrease the diversity of the microbiota, which comprises resistance to colonization that we mentioned earlier, that colonization resistance, right, to infection. We want to resist the infection by incoming pathogenic bacteria, most notably leading to an expansion of C. diff that can cause diarrhea and lead to potentially fatal colitis. Antibiotics have a profound effect on the microbiota that alters the landscape of the gut and can lead to the expansion of pathogenic populations. So that's antibiotics. What about probiotics? Probiotics, there are a lot of these products available created with the intention of inducing a favorable gut balance, But what exactly are they, and do they really help? A 2017 review entitled Probiotics, Gut Microbiota, and Their Influence on Host Health and Disease, quote, "Many probiotics were originally isolated from the gastrointestinal tract and were defined by the Food and Agriculture Organization of the UN as, quote, live microorganisms which when administered in adequate amounts confer a health benefit on the host. Probiotics exert their beneficial effects on the host through four main mechanisms. 1. Interference with potential pathogens. 2. Improvement of barrier function. 3. Immunomodulation and production of neurotransmitters. And 4. Their host targets vary from the resident microbiota to cellular components of the gut-brain axis. However, in spite of the wide array of beneficial mechanisms deployed by the probiotic bacteria, relatively few effects have been supported by clinical data. In this regard, different probiotic strains have effective in antibiotic-associated diarrhea or inflammatory bowel disease, for instance. Specific benefits discussed in this paper include, among others, those elicited directly through dietary modulation of the human gut microbiota. Next, testing. I know testing is super important in the health community. Next, a brief introduction to some of those tools that are used to test or sequence and analyze the gut microbiome. Typically, analysis of the gut microbiome is done by genetic sequencing, and there are a variety of tools for doing so. I'm not going to go into great depth here on my findings, as the details are fairly complex, but I found the following categories of sequencing exist. From a 2021 paper entitled Tools for Analysis of the Microbiome, quote, there are a number of different technologies available to study the microbiome. The list currently includes PCR, or polymerase chain reaction, gene marker analysis, shotgun sequencing, metabolomics, metaproteomics, and metatranscriptomics. The PCR is the earliest form of testing for the microbiome, which many of you have heard of because of its current popularity for testing COVID. Early molecular work in the field was conducted using PCR. The first form of gene marker analysis was 16S RNA sequencing. 16S is a particular portion of a gene sequence that is shared by nearly all bacteria. Basically, ribosomes play an integral role in the creation of proteins. The presence of hypervariable regions in the 16S rRNA gene provides a species-specific signature sequence, which is useful for bacterial identification processes. There is also another form of gene marker analysis called ITS. Next, shotgun sequencing. Shotgun sequencing is the analysis of the entire microbial community. It is based on the extraction of genomic DNA directly from an environmental sample. The great advantage of this method is that it avoids both cultivation and PCR steps, as the DNA is what is being directly analyzed. It can also identify bacteria up to a species level, and the complete or almost complete genome can be assembled. One of the main limitations of shotgun metagenomics is that it does not distinguish the active from inactive members of a microbiome, and thus cannot discriminate those that are contributing to observed ecosystem behavior from those merely present, presumably awaiting more favorable conditions. Next is metabolomics. Metabolomics studies metabolites microbiota produce and how these products interact with both microbiota and host metabolism. These methods often quantify small molecules such as antibodies, antibiotic byproducts, and host or bacterial metabolism intermediates. Metaproteomics. Metaproteomics focus on identifying and quantifying the proteins present within a microbiome. Metaproteomics and metabolomics are both rapidly developing technologies for studying the microbiome. Lastly, metatranscriptomics. Metatranscriptomics is a form of sequencing that focuses on messenger RNA and is considered to provide greater detail on a more specific level than the original gold standard 16S sequencing. The Viome Microbiome Test Kit, which I previously produced a video on, uses this method. For reference on metatranscriptomics, a paper entitled Metatranscriptomics for the Human Microbiome, quote, "In the human microbiome, metatranscriptomics has the potential to provide both basic biological knowledge and epidemiological biomarkers, i.e. high-dimensional diagnostics, for current health status or prognostics for future health outcomes." Most commercially available microbiome tests will either use 16S sequencing or shotgun sequencing, and I found so far that Viome is the one exception that uses metatranscriptomics. For further breakdown of all For further breakdown of all these different sequencing types, see this image from another paper called Microbe 101. From that paper, I will include useful websites about microbiome research. See the list here. To conclude the video, as the scientific community's understanding of the gut microbiome evolves, we should begin to see further use in terms of treatments of disease prevention, reduction, and it will also help drawing connections between what are increasingly known to be the interconnected systems of the gut, brain, and immune system among other systems. In the body, systems rarely work in isolation, but in constant communication with one another. As I've mentioned in other videos, I'm currently doing a commercial test through Viome, which uses metatranscriptomics, and I'm also doing one through a medical provider from Genova for comparison, which uses a variety, I believe, of 16S and metabolomic, as well as some PCR features as well. And we'll do a follow-up with my results so you guys can get a look in what microbial profile looks like, some of the information that's available commercially and through your doctor, and I will be doing kind of a deep dive into my information. I will then turn around and provide that information to you guys along with my analysis and what I plan to do next. That's it for this video, guys. I hope you gained a lot of knowledge and some valuable information from this video. As I've asked, please do subscribe to the video, like it, and I will see you on the next one. Take care, guys. Peace out.